Hello, I'm Peter Midford, a member of the Bioinformatics Research Group at SRI International. This webinar will introduce you to a collection of metabolomics tools available on biocyc.org. BioCyc combines a collection of databases and powerful tools for a variety of bioinformatics tasks. We have 16,800 databases for organisms with sequenced genomes, of which 62 are curated from the literature. The databases integrate genome information with metabolic pathways, gene essentiality data, regulation, and protein features. Our bioinformatics tools support analysis of gene expression data, sequence searching, comparative genomics, and comparative pathway analysis, and the ability to run metabolic models. In this webinar, I will be focusing on BioCyc tools for metabolomics. In this webinar, I will be discussing importing metabolomics data in smart tables, painting metabolomics data onto a metabolic map, calculating pathway perturbation and activation scores, pathway covering sets, visual analysis on the omics dashboard, enrichment analysis, our metabolite translation facility, and metabolite search tools. Most of these tools are integrated with our Smart Tables facility, so we'll start there. I will create a new Smart Table from a file, metabolite measurements from a file derived from a metabolomics workbench study of human cancer. The file contains a list of compounds and their levels in the cancer line SUD-HL1, which normally expresses the oncogene MPM-ALK. The first column contains metabolites. The second column shows metabolite levels measured by mass spectrometry for cells where the oncogene expression is suppressed by the agent CEP26939. The third column shows the metabolite levels under a control condition. Cells were treated with DMSO so the gene is not suppressed. To create the Smart Table, I go to the list of Smart Tables and choose Smart Table from uploaded file. I select the file, and that the file has column headers, and that the first column contains a list of compounds or metabolites. For now, we will focus on the first column, which contains a set of metabolites, so I'll collapse the two columns of measures. The most basic operation with Smart Tables is adding columns showing properties of the objects in the leftmost column. So I'll start with a simple example and add a column of molecular weights. Column of names is selected by default, so all I need to do is choose molecular weight from the Properties drop-down menu. Likewise, I can add another column that contains the INCHI strings for each metabolite. The INCHI strings are rather wide, so I'll collapse those as well. Finally, Smart Table cells can display more than text. Here is a column with structures of each metabolite. In addition to displaying stored properties of compounds, we can apply computational transformations from a different drop-down menu. Other than the separate menu, transformations add columns just like properties. For example, we can compute which pathways a metabolite is involved in. Note that the list of pathways will depend on the current database, so HumanCyc and EcoCyc will list different pathways. This mapping can be accomplished via transformation. Select the transformation pathways of compound. And this gives me a column with the pathways of the compound. I can then click on the plus in the last column, and it will create a new Smart Table that lists the pathways in that column along with matching compounds. Next, I'll show you BioCyc's metabolic map and omics data display. We call an organism's metabolic map diagram in BioCyc the Cellular Overview. To display a set of metabolites on the Cellular Overview, we'll go back to our original Smart Table and collapse a couple of columns. Then I'll go to the Paint Data menu and select Paint Data on Cellular Overview. That brings up the Cellular Overview with the compounds that are in our Smart Table visually highlighted so we can see their relations within the pathway map. we zoom in far enough, it will show the names of some of the pathways. So for example, here in nucleoside and nucleotide biosynthesis, it's a pathway purine nucleotides de novo biosynthesis that contains a few highlighted metabolites. If we roll over a reaction or metabolite, we'll see a tool tip that identifies the entity. If we click on Keep Open, the tooltip window will stay open and we can move it around and in addition, create additional tooltips. The T, R, and P buttons within reaction tooltips let us control how much information is presented. The control panel at the right lets us remove or add back highlighting for multiple sets of metabolites that we might have highlighted at one time. I can remove all highlighting in the operations menu. Quantitative concentrations for one or more time points or experimental conditions can also be painted onto this diagram. For example, if we go back to our smart table, we can then choose Paint Data on Cellular Overview Omics Viewer. I select the DMSO column, choose Absolute, make the data values display linear, and click Go. Next, I can close the summary report. The cellular overview now shows the NMR-derived abundance values of the compounds based on the DMSO column values. The top part of the omics control panel shows a histogram with all the data values in the smart table and the colors assigned to those values. Since the extreme values cause most of the color scale to be assigned to just a few data values, I'll scrunch down the histogram so there's more variation near the bottom of the range. I can zoom in, again using the mouse wheel, and the control panel has a button for removing the data. The cellular overview is more than just the overview of pathways and metabolites. It can also display omics data from a tab-delimited file or several online sources. The data set we have been using has metabolites for two conditions, so we might want to graphically compare the levels of metabolites under these two conditions. I'll start by going back and reselecting the cellular overview, and this will clear any remaining selections or zooming. And now choose the Upload Data from File command. I specify the same file I loaded with the smart table. Then I specify that the file contains compounds. And then I specify the columns 2 and 1 should be displayed. Column 0 is the list of names, and column 2 is the DMSO condition where the oncogene is active, and we'll compare that to column 1 where the gene is suppressed. I select that the values are absolute, and then I click Submit. As before, there is a summary report which I dismiss, and the control panel. I scroll over and zoom into purine nucleotides de novo biosynthesis. And if I roll over DGTP, I see the Keep button, but also an omics button. And this button pops up a graph comparing the abundance under the two conditions. It shows that concentrations are higher when the oncogene is active. Likewise, for GMP and AMP, there is also a difference. And here the colors on the bars make the difference more apparent. The Cellular Overview can also animate time series omics data. I'll load an example set of synthetic human metabolite data. With 5 data points. Specify that columns 1 to 5 are time points for each metabolite in the file, and specify that this is a file of metabolites. Dismiss the summary report. Then I'll just use the play button to start the animation. As the animation steps through the time points, the color for each metabolite is updated. Next, I'll show you several tools that can suggest pathways of interest in an omics dataset. We can generate a table of pathways sorted by their activation levels. One way to do this is to start from the Cellular Overview, and in the Operations menu, load or select some data. I will load the synthetic human data for this because it has multiple time points, and one of the activation statistics only applies for multiple time points. So I'll load data from the synthetic data file. Select compound names. Choose data from columns 1 to 5. Show data as a table, and then select show data as a table of pathway diagrams. Note that the options for show data are checkboxes and not radio buttons. This means you can read data from a file and display it in multiple locations on the Cellular Overview diagram, the pathway diagrams as a pathway collage, or to the omics dashboard. And you could do any combination of these. I will choose to display the top 20 pathways, as this is what we normally do. When I click Submit, the pathway table appears in a new tab. This table lists the pathway name, a diagram with compound levels at each time point, and the 5th column summarizes the enzymes and genes in each pathway. The 3rd and 4th columns show the pathway perturbation or activation statistics. Pathway perturbation scores attempt to measure the overall extent to which a pathway is activated by combining the activation levels of all reactions in the pathway. The 4th column shows a graph of the perturbation scores at each time point for the pathway. The 3rd column shows the differential perturbation score, which is a measure of how much the pathway is perturbed across all time points. It is the difference between the maximum and minimum activation levels across the time points. For a more detailed explanation, see the Pathway Tools User's Guide. Next is our omics dashboard. The omics dashboard shows a high-level overview of all the systems in the cell. On the website, I find the omics dashboard under the Analysis menu. The command launches a page that lets me select data from one of several sources. I'll take the data from the smart table I loaded the cancer study into. Specify that the values are absolute, not relative. And I will turn off molecular weight, basically the I click the Submit button. In a few moments, the dashboard display appears. Here we can see the top-level panels, such as biosynthesis and degradation, and within each panel are finer categories called plots, such as amino acid synthesis or glycolysis. The metabolite levels in each plot is summarized at each condition or time point. For example, in amino acid synthesis, we see the metabolite levels across all amino acid biosynthesis pathways under both the control and treatment conditions. The display shows activity for each metabolite in an amino acid biosynthesis pathway in the small dots. The large dot is the mean of the metabolite levels at each time point, thus depicting the overall level of metabolites in this cellular system. One thing you can do to improve the display is to click on the log scaling button under the display preferences. If we click on a plot, the dashboard drills down to show a more detailed display. Here we have plots for each amino acid biosynthesis pathway. And if I click on an amino acid such as methionine, I can drill down into it and see the levels for each compound involved in the pathway. Another tool we provide is pathway covering. This analysis takes a list of metabolites and determines the smallest number of pathways that will include or cover every metabolite in the list. This may suggest which pathways explain the changing metabolite levels. To do a pathway covering analysis, I'll load a slightly different version of the cancer study file. This file contains the same list of metabolites, but the second column indicates whether the metabolites level has increased or decreased between the experimental and control conditions. The change is indicated by a plus or minus character in the second column. To perform the pathway covering, I will select Pathway Covering from the Analysis menu. Then, when the menu window appears, I select the file and specify a cost function. A cost function is a way to focus which pathways are preferred in the covering set. I will choose the simplest function, the constant function, which expresses no preference for which pathways are considered for inclusion in the covering set. I click Submit, and the analysis displays two tabs. The first lists which metabolites in the file were recognized in the PGDB. The first column is the name in the file, the second is the corresponding object in the PGDB, and the third column indicates that the metabolite is in a known pathway. The pathway covering algorithm ignores metabolites that aren't in any pathway since they can't be covered. The second tab displays the solution set of pathways. The first column names the pathway. The second column indicates the score assigned to the pathway by the cost function, which is 1 for all pathways in this example. The third column contains pathway diagrams. And the fourth column names the metabolites covered by the pathway. The pathway diagram shows where in the pathway the covered metabolite occurs and is colored red or green depending on whether the level is increased or decreased as specified in the file. Enrichment analysis is a statistical technique for finding what a set of metabolites or other objects have in common with one another. That is, do some metabolites in our SMART table belong to specific metabolic pathways at a statistically significant level? For example, enrichment analysis could tell you that a set of metabolites contains more metabolites involved in, for example, fatty acid biosynthesis than you would expect to find by chance. I will load a file that just lists the compounds from the cancer study as a new SMART table. Here's a file that contains just the list of compounds from the cancer study. A list of compounds. To perform an enrichment analysis, we make sure that the compounds column is selected, click on the Enrichments menu, and select Compounds Enriched for Pathways. This pops up a menu with options that control the details of the statistical computation. We'll just take the defaults, although BioCyc provides multiple options for statistical tests and corrections for multiple hypothesis testing. This analysis will create a new SMART table of pathways. Some things to note about this SMART table. The first column contains both individual pathways and pathway classes, which are distinguished by the use of uppercase initial letters for pathway classes, in contrast with lowercase initial letters for individual pathways. The second column is the p-value associated with the statistical calculation, and the column is ordered by p-values. So the pathways at the top are the ones that are most enriched or overrepresented. The last column shows you the compound from the original set that are present in this pathways. Let's delete that match column so we can see the pathways a little better. To delete a column, you select it and use the delete command. We can see here all the pathways that the enrichment analysis has produced, and we can see a lot of them relate to nucleotide biosynthesis. So that is something we've learned. In addition, a number of metabolites are involved in different human amino acid biosynthetic pathways. Next, I will show you BioCyc's search and data display pages. BioCyc contains extensive information on individual metabolites and has extensive search tools. For example, we can search for the compound L-tryptophan in human psych by using the compounds command on the search menu. Search menu provides extensive search options, but we'll start with a simple substring search for L-tryptophan. I can type L-TRY and get a simple drop-down list. Select L-tryptophan from that list. And BioCyc will take me to the compound page for L-tryptophan. This is a multi-tabbed page where each tab shows a different dimension of information. We can switch tabs to see such as any reactions the compound regulates, growth media containing the compound, and reactions the compound is involved in. From that page, I can go to pathways involving the compound, such as L-tryptophan degradation via the tryptamine pathway. This takes us to the page for that pathway, and from the pathway diagram, we can examine the enzymes in the pathway, such as L-tryptophan decarboxylase. This enzyme catalyzes multiple reactions, and we can scroll down in the decarboxylase reaction and select it. This takes us to the reaction page. We can also show this reaction on the Cellular Overview. Of course, there are other ways to search for compounds. For example, if we're looking at mass spectrometry data, we might want to look at metabolites with a particular monoisotopic mass. BioCyc supports that search and allows the specification of a tolerance around the specified mass. And this particular search mass takes us back to L-tryptophan, and we see both isomers of the compound are returned. Finally, I'll show you BioCyc's metabolite name translation tool. BioCyc provides a batch tool for interconverting metabolite names and identifiers from different databases. Metabolite translation service, you'll find a tool. I'll take a list of metabolite names and report the corresponding BioCyc names. You can type in the list in the box or load a file. For simplicity, I'll use the built-in example set, which loads with this button. Because the names can come from different databases, the list consists of identifiers prefixed by their database IDs, such as PubChem 125 or KEG C00001. IDs from different databases that identify the same compound should be on one line and separated by tab characters. Then click the Send button. The result page lists the name submitted, whether the translation was successful, and the resulting BioCyc metabolite name, followed by other synonyms. We hope you've enjoyed this tour of the metabolomics tools provided by BioCyc